Hi, I'm here today to simulate to you mitosis using a set of chromosomes. So if we look down here, I am starting with a cell with a diploid number of 4. So I have 4 chromosomes inside of my nucleus. Here's my nucleus with sort of this white string. Here is my cell membrane out here in the red string. And then these are to represent, you guys know that I'm not very crafty, these are to represent like mitochondria and other organelles, Golgi apparatus and things like that that the cell might need. All right, so to back up in the cell cycle, before we get to mitosis, one of the things that needs to happen is DNA replication. That happens in S phase, or we copy all of these chromosomes. So at the beginning of replication in S phase, we are going to basically make copies of all of these sort of chromosomes. So we go from 4 chromosomes, that's our diploid number, our starting number, to 8 potential chromosomes inside of our nucleus. Now I want to show you mitosis. So skipping down to mitosis, the first phase is prophase. In prophase, all of these chromosomes are going to find their identical copies and link at the centromere. So you see how they carry the same sort of genes and the same colors. So each of these like sets will find their sort of partners and they will link. And they should be identical, right? Because we've also checked and proofread our DNA in G1. Now all of our sisters are connected at this centromere. Remember, that's a sticky protein, and we can move on to prometaphase. In prometaphase, the nuclear membrane disappears, so that's gonna go away from our cell. And for right now, I don't have microtubules. My hands are the microtubules. And so imagine that all of these microtubules grow out and one side is gonna connect to one half of the sister chromatid. The other microtubule will connect to the other side of the sister. They link around the protein at the middle so that they don't disrupt or mutate any of the DNA. Now, once the nuclear membrane is gone, then the chromosomes are hooked up to the mitotic spindle. In metaphase, we line up all of our sisters so that they are in a line in the center of the cell, like so. So those microtubules kind of shift them around until they're on a line. We run the metaphase checkplate or checkpoint where the enzymes will check to make sure all of the sisters are hooked up correctly. The next phase is anaphase, and anaphase, all the sisters separate. So all your sister chromatids separate. And as they separate, they kind of are pulled to the opposite ends. So that leads us to telophase. And telophase, for the chromosomes, half of them will go to one end of the cell, half will go to the other. And in telophase, your nuclear membrane is going to return around those chromosomes. Remember, we started with a diploid number of 4. So at the end of telophase, we should have 4 chromosomes that are identical in each of our cells. Finally, cytokinesis is where the cell pinches off. So the rest of the cell contents, like about half of the mitochondria, Golgi apparatus, will go one way and half of it will go the other way. And the cell pinches off to create 2 identical daughter cells. So in mitosis, we start with 1 cell that is diploid and we end with 2 identical diploid daughter cells. Thanks for watching this simulation, and I will see you next time.